குட் மார்னிங். இன்று என்சைம்ஸ் பற்றி பார்க்கலாம். Introductions of enzymes and their properties. Enzyme is a substance that acts as the catalyst in living organisms, regulating the rate at which the chemical reactions proceeds without substance being altered in the process. The biological process that occur within all living organisms or chemical reactions, most of which are regulated by the enzymes. Without enzyme, many of these reactions would not occur at a perceptible rate. Enzyme catalyzes all aspects of the cell metabolisms. This include digestions of the food, large nutrient molecules such as the proteins, carbohydrates, and the fats are broken down into the smaller molecules. Conservations and the transformations of the chemical energy, constructions of the cellular macromolecules from smaller precursors. Many inherited human diseases such as the albinisms and the phenylketonuria results from the deficiency of the particular enzymes. Enzymes also have valuable industrial and medical applications. The fermenting of wine, leavening of bread, and curding of cheese have been practiced from earliest times. Since then, enzymes have the assumed increasing importance in the industrial process that involved organic chemical reactions. In medicines, enzymes are used for killing disease-causing microorganisms, promoting wound healing, and diagnosing certain diseases. Properties of the enzymes. Enzymes are proteins that initiate and speed of biological reactions. pH affect the enzyme activities. Each enzyme is most active at a specific pH. For example, pepsin has a pH optimum of 2, whereas uh, tri- trypsin has a pH optimum of 8.5. Most internal enzymes function near neutral pH. Enzymes have active sites that engage in biological activities and can accelerate reactions in either directions. Enzymes are unstable molecule soluble only in dilute glycerols, sodium chloride, and dilute alcohol. Enzymes are temperature sensitive and are most active at optimal temperatures. Although all enzymes are proteins, not all proteins are enzymes. Enzymes lower the activation energies of the substrate molecules, allowing biochemical reaction to occur at a body temperature 37 degrees centigrade. Catalytic properties. Enzymes are biological catalysts with a high catalytic efficiencies. A small amount of the enzymes can catalyze large quantities of the substrate into a product. Enzymes accelerate reactions while remaining unaffected by the reaction they catalyze. Enzyme specificity. Enzymes are highly specific, meaning that a particular enzymes catalyzes only one type of the reaction. For example, uh, sur- uh, surcase executely catalyze the hydrolysis of the sucrose. Characteristics of the enzymes. Being protein, enzymes exhibit all the properties of the proteins. They have specific isoelectric points where they are least soluble. Enzymes can be denatured by the change in pH and temperature. High reaction rate. Enzymes catalyzed reactions are 10 power 6 to the 10 power 12 times faster than uncatalyzed reactions. Medullary reaction conditions. Enzymatic reactions occurs at relatively low temperatures below 100 degrees centigrade, atmospheric pressures, and nearly neutral pH. Capacity for regulations. The catalytic activities and the amount of enzyme synthesis are highly regulated. Regulatory mechanisms included allosteric control and the covalent modifications. Greater reactive specificity. Reactions catalyzed by the enzymes are highly specific. Two types of the specificities are observed. One, absolute specificity. The enzymes catalyzes only one particular reaction. Example, urease acts only on urea. Group specificity, the enzymes act as on a group of the substance with a particular type of the chemical in linkage. Enzyme acting on, example, enzyme acting on starch, dextrin, and glycogen, which all have the same glycosidic linkage. Classification and nomenclature of the enzymes. Enzymes, oxidoreductase, transferase, hydrolysis, lylase, isomerases, uh, ligases. Types of enzyme. Oxidoreductase. These catalyze the oxidations and reduction reaction. Example, pyruvate dehydrogenases, which catalyze the oxidations of the pyruvate to the acetyl coenzymes A. Transferase. These catalyze the transfer of the chemical groups from the one compound to the another. Examples, transminase, which transfers an amino group from one molecule to the another. Hydrolysis. These catalyze the hydrolysis of the bond by adding water. Example, pepsin, which hydrolyze peptide bonds in proteins. Lysis. These catalyze the breakage of the bonds without hydrolysis or oxidation. Example, aldase in glycolysis, which is split fructose 1,6 by phosphate into the glyceraldehyde 3 phosphate and dihydroxy acetone phosphate. Isomerase. These catalyze the formations of an isomers of the compound. Example, phosphoglucomatase, which converts glucose 1 phosphate to the glucose 6 phosphate. The phosphate group is transferred within the same molecule during glycogenesis. Ligase. These catalyze the associations of the two molecules. Example, DNA ligase, which join two fragments of DNA by forming a phospho diester bond. Biochemical properties of the enzymes. Uh, Oxidoreductase catalyzes oxidation reductions reactions where electron transfer from one molecule to another. Transferase help in the transfer of the functional groups among donor and acceptor molecule. Hydrolase, hydrolytic enzymes that cleavage bonds by adding water. Lysis, add remove the water, carbon dioxide, ammonia across double bonds or eliminate these two from double bonds. Isomerase catalyze structural shifts within a molecule, changing its shape. Ligase catalyze ligations process joining two molecules together. Thank you.